Students, good afternoon all of you. Our next topic discussion on artificial hybridization. See, from this chapter, it is very important question. So compulsory they will give this question. Okay. So listen carefully. Artificial hybridization. The word itself saying the meaning. Artificial hybridization. So artificial means what? With the help of with the help of plant breeders. Hybridization means what? Breeding process. See here. So it is one of the important technique for improvement of various kinds of crops. Okay, that is called artificial hybridization. In this one, so the pollen grains in this technique, pollen grains from a desired plant are collected and placed on desired plant stigma. That is called artificial hybridization. Okay, see, so pollen grains, the plant which is growing healthy. That plant is must be disease resistant variety and should be grown in any climatic conditions. From such kind of a plants, plant breeders collects pollen grains. In this technique, they will collect pollen grains of desired plant that may be disease resistant variety. So any climatic conditions it can grow. So it can give excess amount of crop. Such type of plants collected from those plants, pollen grains are collected and placed on the stigma. So that stigma means placed on the female flower. So the female flower also should be superior variety. See here, so this is the stigma. Yesterday we have seen that process. That is natural hybridization. So it is artificial hybridization. So from another plant. So the anther, from the anther, this is the anther. From the anther, pollen grains are collected and placed on the superior variety of female flower. So generally pollen grains are placed on the stigma. There pollen grains are germinated, further process takes place. Okay, so this is called artificial hybridization. So in this artificial hybridization, the main step, we have to protect the stigma. We have to safeguard the stigma from the contamination of pollen grains. From the contamination of pollen grains means what? Unwanted pollen grains, there is a chances to land on the stigma. Yesterday we discussed that. Often there is a incompatible, mismatching, wrong type of pollen grains, there is a chances to land on the stigma. So to stop this, we have to safeguard the stigma from contamination of unwanted pollen grains. For that, we have to follow some techniques. Okay, so how we will protect this stigma? To protect this stigma, we have to achieve two methods, two techniques. Okay, artificial hybridization method is carried by two techniques or two methods. Okay, artificial hybridization technique is achieved by two methods. One is emasculation. Another one is bagging. Emasculation and bagging. So first of all, let us see the emasculation. In this technique, from the desired flower, desired variety of flower we have to select. Desired variety means what? That plant should have superior qualities such as disease resistant variety, genetically high characters. It should grow any climatic conditions. It must give superior variety of fruits. So those charactered plant we have to choose. Such type of a character plant, we have to choose a flower. So from that flower, the male reproductive parts we have to be cut. We have to be removed with the help of forceps or scissors, whatever may be. So from that flower, male reproductive parts we have to remove with the help of scissors or forceps. This is called emasculation. So the female reproductive parts, that is what? Stamens. The complete stamen we may be removed, or else only anther we can be removed. Then from that particular flower, from the anther to stigma, that is self-pollination does not take place. Self-pollination does not take place. If self-pollination occurs means, so it gives less superior varieties. That's why. So from the superior variety of plant, we have to choose the flower. From that particular flower, male reproductive parts, we have to be removed, either stamen or anther. Whatever it, if we will remove here, anther won't be there. So if we will leave this, only stamen, if we will cut, only stamen, if we will cut, it is also okay. This technique is called emasculation. This is called emasculated flower. Okay, next, bagging. The word only saying the meaning, bagging, bagged. So the emasculated flower needed to be bagged with transparent suitable size of stigma. Suitable size of stigma, transparent bag, we have to take it and cover it with transparent bag. So when we have bagged, the date, time, we have to be mentioned. Date, time, month, we have to be mentioned. So this technique is called bagging. Very simple. So the emasculated flower, we have to be covered with transparent bag. That's maybe a polythene cover or any transparent cover. So that cover should be suitable to this size. Suitable size of transparent bag, we have to be covered. After bagging, we have to record date, time, month. Okay, that is called bagging technique. So after bagging, for example, today we have bagged a specific emasculated flower. After 15 days, 20 days or 1 month, this stigma attains maturity. Attains means gets maturity. The stigma receptive. When the stigma is receptive means the stigma is mature at that time. So this bag we have to be removed. The bag we have to be removed. And from the desired plant, pollen grains we have to bring and place on the stigma. Pollen grains we have to be placed on the stigma. Then again, the bag is needed to be rebagged. Okay. So if we will not rebagged, what happens here? There is another species of pollen grains chances to land. That's why until finishing these pollen grains germination, we have to be bagged, rebagged. We have to be rebagged. Again, the bag we have to be tied with the threads. So these specific pollen grains are germinated, pollen tube grown, that pollen tube reached to the pistil, their fertilization takes place, seeds are produced. Those seeds are called hybrid variety seeds. Those seeds are also called superior variety of seeds. This technique is called artificial hybridization. So artificial hybridization is achieved by two methods, emasculation and bagging. Okay, so why we needed to bag? We have to safeguard the stigma from the contamination of pollen grains. Contamination means what? Unwanted species of pollen grains, there is a chances to land. To protect the stigma, we have to be covered with transparent suitable size bag. So if this flower is bisexual, emasculation and bagging technique both are required. If the flower is unisexual, emasculation does not required, only the bagging is required to produce superior variety of crops. This is called artificial hybridization. This question carries for 3 marks, definite question. Okay, ma? That's all. Like this way, diagrammatic representation also, if you give, you will get good impression. Okay, that's all. Double fertilization. Double fertilization. See, the double fertilization is discovered by Navasin. In Fritillaria and lilium plant. Double fertilization. So since last year we are discussing about the double fertilization, how double fertilization takes place in flowering plants. Already we studied, but specifically under this heading here we will see that. So double fertilization. So first time in flowering plants, Fritillaria and lilium, these are flowering plants. In flowering plants, double fertilization observed by the scientist Navasin. Okay, so how double fertilization takes place, that is what here we will see. It is also important topic. So 1 year they will ask double fertilization, 1 year they will ask artificial hybridization. These two questions are exchanged, they will ask. March month if they will ask, this is June they will ask artificial hybridization, like that successively they will ask this question. Okay, so in this one, so the first male gamete are haploid nuclei. Male gamete or haploid nuclei. So already you know very well how haploid nuclei are formed. So when pollen grains are landed on the stigma, at that moment pollen grains are germinated. The germinated pollen grains are produced two types of male gametes. Those two male gametes are reached to the pistil and fertilization takes place. In those two male gametes, one male gamete or haploid nuclei is fused with, is entered into one of the synergids. Cytoplasm of the synergids, there it is fused with egg cell. So haploid male gamete and a haploid female gamete both are fused together and produced zygote. It is observed by the scientist Strasburger. So this is called zygote formation. So this is called first fertilization. So this is what observed by the scientist Strasburger in monotrapa plant. So when we will start objective discussion, in that objective discussion, these bits we will get. Okay, so see, double fertilization, he is first fertilization Strasburger, he observed in the plant monotrapa plant. So this is called first fertilization. It is also called syngamy. After syngamy, so one more male gamete. It is also haploid. It is fused with secondary nucleus, that is polar nuclei. So in the embryo sac, polar nuclei is present. Immature form of secondary nucleus is called polar nuclei. So polar nuclei gradually matured and fused, then it becomes secondary nucleus. So 2n state. So this 2n state and male gamete both are fused. So one more male gamete is entered into the center of the embryo sac. There it fuses with the secondary nucleus and forms triploid nucleus. This triploid nucleus is gradually called primary endosperm nucleus. This primary endosperm nucleus gradually develops into endosperm. Since two sets of fertilization occurs in embryo sac, it is the characteristic feature of the flowering plants. Angiosperms. It is the characteristic feature of the angiosperms. It occurs only in angiosperms. Okay, since two sets of fertilization, it is the first set of fertilization, it is second set of fertilization. Since the two sets of fertilization takes place in embryo sac and leads to formation of triploid nucleus, it gradually turns to be primary endosperm nucleus. Primary endosperm nucleus undergoes a number of mitosis divisions and gives rise to endosperm. Okay, that's all. So this is double fertilization. Okay, that's all. Next here. See, after double fertilization, what are the changes takes place? See here, it is everybody knows that. So the zygote undergoes mitosis divisions and gives rise to embryo. This is one. Next, this primary endosperm nucleus. Actually, I said here only complete information. Actually, here triploid nucleus. So this is called triple fusion. 1 mark definition. So triple fusion. Define triple fusion. 1 mark question. The fusion of male gamete with the secondary nucleus is called triple fusion. So the triple fusion results in primary endosperm nucleus. The primary endosperm nucleus undergoes number of divisions, number of mitosis divisions, and gives rise to endosperm. This endosperm provides nourishment to the developing embryo. In some of the seeds, such as GMAs, some of the seeds such as GMAs means what? Monocot seed. In monocot plants, endosperms stores the food material. Endosperm stores the food material. See that only. So primary endosperm nucleus undergoes mitosis divisions and gives rise to endosperm. This endosperm provides nutrients, nourishment means nutrients to the developing embryo in the seed. See here, so some seeds such as GMAs means what? Corn plant, mekejoli. Okay, in GMAs, endosperms stores the food material. In some of the seeds such as dicot seeds, so this endosperm is completely utilized by the embryo. Then instead of food storage endosperm, it occurs in cotyledons. In monocot, endosperm stores the food materials. In dicots, cotyledons stores the food material. So that food material is supplied to the developing embryo during seed germination. So this is the double fertilization. This entire process is observed by the Navascene in plant, Fritillaria and Lilium plant. So in exams from this topic, 3 types of questions they will ask. So what is double fertilization? Where does it occur? What is triple fusion and the importance? Like this way. Double fertilization means these 2 you have to write. Where does it occur? In flowering plants. Since the 2 sets of fertilization takes place in flowering plants, it is the characteristic feature of angiosperms. Next, what is the triple fusion question they will ask? So the fusion of male gamete and the secondary nucleus is called triple fusion. What is the importance of triple fusion? So the triple fusion results primary endosperm nucleus formed. As a result of primary endosperm nucleus, endosperm is formed. The endosperm provides nourishment to the developing embryo. These are all the things you have to be mentioned. Next topic, post-fertilization structures and events. So after fertilization, what are the changes takes place in flowering plants? That is what in this session we will see. See, after double fertilization, the flower undergoes various changes. Those changes are, first one is development of endosperm. Second one is development of zygote till the formation of seeds. Next, development of seeds. Development of seeds. Next, development of fruits. Development of fruits. So these are all the changes takes place in flowers. These are called post-fertilization structures and events. If we will discuss these are all the events, our second chapter finished. So before that, first of all, let us talk about development of endosperm. Thereafter, these headings we will see that. See here, primary endosperm nucleus undergoes mitosis divisions and gives rise to endosperm. So the endosperm is always triploid. How does endosperm formed in flowering plants? The question, 2 marks question. Endosperm is formed as a result of triple fusion, so that it is a triploid. In flowering plants, the development of endosperm begins earlier, that is before the development of embryo. Before the development of embryo. See here, it is seed coat. It is seed coat. Embryo, seed coat, endosperm. So in flowering plants, the endosperm development occurs earlier than the formation of embryo. See, so before the formation of embryo, the endosperm is formed because this endosperm provides nutrients to the developing embryo. This is called development of endosperm. Generally in flowering plants, the endosperm development is 3 types. 3 types of endosperm is formed. Types of endosperm. Types of endosperm. In flowering plants, 3 different types of endosperm is commonly formed. Those 3 types of endosperms are, first one is free nuclear endosperm. The second one is cellular endosperm. And third one is halobial endosperm. Halobial endosperm. See, free nuclear endosperm. How does free nuclear endosperm is formed? That is what in this session we will see. So in this one, primary endosperm nucleus undergoes mitosis divisions and forms a number of nuclei. These are all the nuclei are settled at the periphery. Periphery means what? Outer side. These are all the nuclei are settled at the periphery, leaving the central large vacuole. That is called free nuclear endosperm. So free nuclear endosperm commonly formed in maize plant, rice plant, sunflower plant, coconut, etc. In those plants, free nuclear endosperm is formed. So, in these plants, maize, rice, sunflower, free nuclear, you may not understand clearly because maize, rice, sunflower, clearly you may not observe. It is clearly observable in coconut plant. In coconut plant, in the tender coconut, free nuclear endosperm is a best example. In the tender coconut means what? So in the tender coconut, we will drink water, coconut water. So the coconut water contains many nuclei. That is called free nuclear endosperm. Free nuclear endosperm. When primary endosperm nucleus undergoes mitosis divisions and produces number of nuclei. Okay. Okay, that's all. That is called free nuclear endosperm. For the free nuclear endosperm, a best example is the tender coconut. In the tender coconut, coconut water is a best example for free nuclear endosperm. So the coconut, this is coconut. So here it leaves a large central vacuole. It is filled with a water. So the coconut water is surrounded by a white kernel. So that white kernel is the best example for cellular endosperm. Okay. So around the coconut water, the white layer, white wall is called kernel. It is a cellular endosperm. Okay. So that is a cellular endosperm nucleus. Only coconut water, it contains number of nuclei. That is free nuclear endosperm. So for the cellular nuclei, for the cellular endosperm, another meaning also is there. Another type of explanation also is there. What is that? So a thick wall is formed. A thick wall is formed after the division of primary endosperm nucleus. Primary endosperm nucleus divides by number of divisions and forms a thick wall. So the formation of thick wall is the best example for the cellular endosperm. So this thick wall contains number of cellular endosperms. So this thick wall formation is best example for cellular endosperm. So this type of endosperm commonly seen in Datura plant. Petunia, Datura, Petunia, and balsam plants. Okay, so you maybe remember white kernel around the coconut water is called free nuclear endosperm. If not possible, it is also a thick wall formed after division of primary endosperm nucleus. So the thick wall is a best example for cellular endosperm. So this thick wall formation commonly seen in Datura plant, Umbetta plant, Petunia plant, and balsam plant. Last and final, helobial endosperm. In this helobial endosperm, this is called primary endosperm nucleus. Primary endosperm nucleus undergoes transversely divided. Primary endosperm nucleus divides transversely and produces large micropylar chamber, and small chalagel chamber. That means what? Two unequal divisions. Unequal division of primary endosperm nucleus and gives large micropylar chamber towards micropylar region and small chalagel chamber towards chalagel and in embryo sac. That is called helobial. Helobial means unequal division of primary endosperm nucleus. It is seen in Asphodylus eremourus plant. Eremourus plants, Asphodylus and eremourus plant. In them, this kind of halobial endosperm we can be seen. Depending on the development of endosperm, the endosperms are three types. Those are free nuclear endosperm, cellular endosperm, and halobial endosperm. These are three types of endosperms commonly seen in different types of flowering plants. Okay, so this is the explanation about the development of endosperm. Next, development of zygote till the formation of seeds. That the second stage and remaining two stages we will see in next class.